Good evening. Good evening to you all. Hello, am I audible? Hello, can you hear me? Am I audible? If yes, you can give me a thumbs. Hello, hello. Okay, very good evening to you all. This is Amala teacher from GoScore Learning, and this is the fastest edtech in GCC. My dear students, in the previous class, we have discussed about chapter 9, that is biotechnology principles and its process, the first portion, right? So, what we have discussed in the previous class, can anyone comment in the class? Anyone have any idea on what we have discussed in the previous class? Yes, it was about biotechnology, right? So, what is biotechnology? As I have mentioned, it is the integration of technology with biology. Earlier, we have practiced biotechnology. As example, we can consider the curd, right? It is actually a biotechnological process, but we doesn't know that it is a biotechnological process, right? It involves the microorganism, and the pure milk will be converted into curd with the help of a microorganism. So that technology have been modified and it is being used in different purposes for the what we can say, for the purpose of the human needs. Okay, we can say biotechnology, the final product will be very much useful for the human beings. Okay, so that's all about the simple term for biotechnology, and you know there is a definition for biotechnology. My dear students, you have to study that definition for biotechnology. Okay, so after that we have to take into consideration about what are the different tools used for biotechnological processes, or to make a recombinant DNA, what are the different biotechnological tools? Okay, we have already studied about that, right? So what was that? It was about enzymes. Did you remember that? It was about enzymes. Okay, what enzymes we have studied? We have studied about restriction enzyme, right? Restriction enzyme. Did you remember the term restriction enzyme? Yes, what is restriction enzyme? The enzyme which is used to cleave the DNA is known as restriction enzymes. And also, dear students, you remember that there are two different types of restriction enzymes, right? It is endonuclease and exonuclease, right? What is endonuclease? If the DNA is cleaved within the DNA, okay, within the DNA, then that kind of enzymes are known as endonucleases, right? So what are exonucleases? Exonucleases are the nucleases that are cleaved out of the outer side of the DNA. So that are the different types of the nucleases, or we can say restriction enzyme used for the cleavage of DNA. Okay, so DNA is cut into different fragments. After cutting, this have to be recombinated with the other what we can say, vector DNA. So we have learned the term vector also in the previous class, right? What is vector and what are, we have studied an example of vector with PBR 322, right? If you are considering PBR 322, we have learned about the ampicillin resistant gene side, then tetracycline resistant gene side, ROP gene side, then origin of replication, and also the different sites for the cleavage for restriction enzymes. Okay, so these are the tools for recombinant DNA technology. Today, my dears, will be discussing with processes of recombinant DNA technology. What are the different processes involved in recombinant DNA technology? Okay, that is the first step. Okay, once again we can think of what are the tools which have to be used for DNA recombinant technology. It is of course the restriction enzyme. You have to keep in remember about restriction enzyme, and also you have to keep in remember about the vector molecules that are present. Okay, that should be there in your mind. Also, you have to keep in remember about the host organism where it has to be introduced. Okay, that are the three important tools that you have to keep in remember. So today we will be discussing about the processes of recombinant DNA technology. Okay, so let's start with the first process of recombinant DNA technology, that is isolation of the genetic material, that is DNA. Okay, you know, we can consider a cell. Okay, this is a cell, right? Consider this as a cell. Okay, so we can consider that as an animal soul. So we can say at the middle, what is there? Nucleus will be present. What is present? Nucleus will be present. So we can write that this is a cell or plasma membrane, right? What is this? This is plasma membrane. Okay, so inside the cell, what is present? Nucleus is present. What is present? Nucleus is present. Okay, so inside nucleus, what is present? This DNA is wrapped together. Okay, as chromosomes with histone proteins. Okay, it can be seen only in which phase? Mitotic phase. Okay, so we can say inside the nucleus what is present? In simply we can see that there is DNA. What is present? DNA is present. So this is about a cell. From the cell we have to obtain the DNA. We have to take out the DNA for the further processes. Right? So what is the first step? It is the isolation of the genetic material. What is the genetic material? Genetic material is the DNA. Okay, so how we can isolate the DNA? So to get pure DNA, what we have to get? We have to get pure DNA. There shouldn't be any kind of in impurities. Okay, so we can say they should be free from other macromolecules. The bacterial cells, plant cell or animal tissue are treated with enzymes. Study what are the different enzymes involved. Okay, what are the different enzymes? First one, it is lysosome. For bacteria, see, the plasma membrane have to be broken. First, the plasma membrane have to be broken. Then the nucleus have to be broken. If from the nucleus, what have to be taken out? DNA have to be taken out, right? So, in order to destroy the plasma membrane, what has to be used in bacteria? Lysozyme, cell wall. Okay, cell wall, not plasma membrane. Above the plasma membrane, there is cell wall in the case of bacteria and plants. If you are looking into the fungus also, there is cell wall. So, first of all, what we have to break, if it is a plant cell. Okay, we have considered this as animal cell. If it is a plant cell, see, there is a cell wall. Okay, right? There is a cell wall. Inside the cell wall, what is present? Inside the cell wall, a plasma membrane will be present. Inside the plasma membrane, there will be a nucleus towards the peripheral side since vacuoles are present. Okay, so see, this plasma membrane have to be destroyed to obtain the DNA. Okay, to obtain the DNA which is present inside the nucleus. Okay, which is present inside the nucleus. Understood? So first of all, we have to remove the cell membrane, cell wall, not cell membrane, cell wall. Okay. Then we have to destroy the plasma membrane, then the nucleus. Out of the nucleus, we have to take out the DNA. So how the cell wall composition in bacteria is being removed? It is by lysosome. Okay, it is by lysosome, not so, it is lysozyme. Okay, so in the case of plant cell, how can it be destroyed? It can be removed, it can be treated. The plant cell have to be treated with cellulase. What is cellulose? What is cellulose? It is the plant cell wall composition, right? The plant cell wall is made up of cellulose. So, in order to break down the cellulose, what we are using? Cellulase is using. What is using? Cellulase is using. Then, next consider the case of fungus. Okay, what is the cell wall composition of fungi? Fungi. What is the cell wall composition? It is chitin, right? Chitin. So we are using chitinase as the enzyme to remove the cell wall. Okay, clear, understood. So once the cell wall is removed, the other molecules will be also treated with different chemical molecules. Let's see what are the chemicals that are being used to take out the DNA from the cell. Okay, so the cell is broken to release DNA along with other macromolecules. Okay, so once the cell wall is taken out and the plasma membrane is removed, the DNA will be taken out and along with the macromolecules. Okay, what are the different macromolecules that is present along with DNA? It is RNA and protein, then polysaccharide and lipids. Okay, next target is to remove this RNA, then what? Protein, polysaccharide and lipid. Let's see how it can be removed. Also, dear students, we have to understand, we have already studied in molecular basis of inheritance that always DNA is intertwined with proteins known as histones. Okay, how DNA is turned to be a chromosome. How DNA is turned to be chromosome? DNA, then it is intertwined to form a structure called a nucleoid structure. Nucleosome structure. Nucleosome structure will give the solenoid structure, 300 nanometer structure. Then it will be turned to a 600 nanometer structure. Then it will be turned to a 1400 nanometer structure. That is a chromosome, right? So here, how it was arranged. How it was arranged? The DNA was wrapped around the positively charged molecule, that is histone protein. So keep in remember that genes are always intertwined with proteins known as histones. Okay, clear? So next, what is the target? What is the target? We have to take out the DNA from the cell. Okay, let's see how the DNA can be taken out with the other molecules. Okay, DNA which is present along with other molecules can be taken out. Okay, so next the treatment is, next what is present? RNA is present. Okay, once it is taken out, RNA will be present. So how RNA will be treated? RNA is removed by ribonuclease. Okay, how it is taken out or how it is removed? See, what if you are taking the condition now, the DNA is present along with RNA. Okay, then protein is present. We have to remove all these things. Protein is there. Then different molecules. Polysaccharides will be there. Okay, polysaccharides are there. Then what is present? Lipids are present. So all these things have to be removed. All these things have to be all these have to be removed in order to get a pure DNA. What is the condition? We have to obtain a pure DNA. Now the DNA is with this lipids, polysaccharides, RNA and protein. So in order to remove the RNA, what we have to do? We have to treat it with ribonuclease. Okay. Then proteins are present. I have mentioned that here protein is present. Okay. This protein, how the protein can be treated? Protein can be removed by the help of the enzyme called protease. Dear students, you have to study what are the different enzymes used to remove all these things. Okay, then other molecules it is removed by appropriate treatments. You just want to study how it can be, what steps have to be taken to remove. It can be removed with the different chemical molecules. Okay, so next process we get DNA, right? Now we have a DNA in our test tube. So we can consider we are isolating it in a test tube. Okay, now the DNA is present in the test tube. We need this DNA in a visible form. Okay, actually when we are isolating DNA, it is not in a visible form, but when we are adding the chilled ethanol into it. Okay, chilled ethanol means cooled ethanol. Okay, so if this chilled ethanol is present, then what happens? This purified DNA precipitate out as a collection of fine threads in the suspension. So when if you are isolating, this is the isolation. Okay, DNA isolated. This is the DNA isolated suspension. Okay, if this is DNA isolated suspension, when we are adding suspension, okay, suspension, when we are adding the chilled ethanol into it, we are adding the chilled ethanol into it. Okay, what we are adding? We are adding chilled ethanol. So what happens here if we are adding chilled ethanol into it? The DNA which is present inside this will be precipitated as white threads. Okay, it will be precipitated as white threads. So when it is precipitated, we can take it out for the further steps. Okay, this is how the DNA is being isolated. That is the first step, isolation of desired DNA. Isolation of desired DNA. How DNA is being treated, you have to remember about a structure of cell, that is, it is made up of a cell wall. First, the cell wall have to be removed. How the cell wall in bacteria is removed, how the cell wall in plants is removed, how the cell wall in fungus is removed. That is the 3 points you have to keep in remember. Okay, next, how the DNA along with different macromolecules have been treated to get a pure DNA. So two conditions have to be applied here. Okay, fine, understood, clear. Okay, so next we will be moving into the next step of the process, that is, we have isolated the DNA as thread-like structure, right? Thread-like structure in a suspension, So this thread-like structure DNA will be taken out. Okay, then what happens? It is treated with restriction enzymes. Okay, we have studied what is restriction enzymes, right? They are the enzyme which is used to cleave the DNA. So this is an entire DNA of an organism. So we doesn't want this entire complete DNA, right? Only a small portion of DNA have to be taken from the organism. So what is done here? This DNA will be treated with restriction enzymes. Okay, after using restriction enzyme, we'll get an extracted DNA, an exact DNA portion. Okay, we'll get an exact DNA portion. Since DNA is in double-stranded structure, we'll draw it in double-stranded structure. Okay, fine. So see, restriction enzyme digestion. Okay, restriction enzyme digestions are performed by incubating purified DNA with a restriction enzyme at the optimal condition. Given an optimal condition, what is being practiced here? A restriction enzyme is used here to cleave the DNA and the desired target DNA is ready. That is the second step. Okay, next step. What is the next step? Isolation of derived DNA fragments. Okay, next step is to isolate the accurate DNA fragment. For that, what we are practicing, we are practicing the step called agarose gel electrophoresis. Every time the board seems to ask this as a question. Okay, what is agarose gel electrophoresis? Okay, what are the steps or what is involved in agarose gel electrophoresis? So we get an DNA, right? We'll get a DNA. We'll get a pure isolated DNA which is being treated with restriction enzyme desired DNA. That is the portion of desired DNA. Now we need an accurate DNA and that has to be copied next. Okay, so you have to think, you have to remember that in your vision. Okay, see, the cell is taken, then the outer surface, that is the cell wall is removed. After cell wall, what is present? Plasma membrane. It is also being treated with some chemicals to remove this plasma membrane. Then into that, what is present? DNA. The nucleus is present. Nucleus will be containing the DNA, protein, etc. Okay. From there, we have to get the DNA. Once the DNA is isolated, what we are using? Restriction enzyme is used to get that particular sequence of DNA. Okay. After that, it has to be again treated to get that particular specific DNA. Okay. So, here what practice is being used? Agarose gel electrophoresis will be used. Next, we have to study what is agarose gel electrophoresis. Okay, it is a method to isolate and separate the DNA according to its molecular weight. Okay, so if you are considering and this is the diagram which is given in your textbook. Agarose gel electrophoresis. So you can find here the writings as 1, 2, 3. Okay, so this is the first well. This is the first well. Okay, this will be the second well. Okay, second well. Well, okay, that means the DNA have to be poured into this well. Then it will be moved accordingly. Okay, so this is the first well, this is the second well, this is the third well, and this is the fourth well. 4 wells will be prepared. Okay, we are actually, there is an electrophoresis plate. We are pouring the agarose into it. Gel we are pouring into it. We are preparing the gel. Okay. During the preparation, the well also have to be prepared. Okay, fine. So how this well is or the gel is prepared? It is made with agarose. Okay, agar, which is taken by a seaweed. Last year, 11th standard, first, second chapter, you have studied about a seaweed, which we can take the agar from it, right? Comment it, okay? So there are, with the agar, we are preparing the agarose at first. Okay, so we can consider a plate here. Inside the plate, we are adding the agar. Okay, so when we are adding the agar, we will get different wells as like this. Okay, how many? 4, 5 wells or 6 wells will be present in an agarose gel unit. Okay, so inside this well, what we will take or what is the next process? Okay, this next gel is, see, this is inside an apparatus, right? This apparatus will be connected with electricity. Okay, anode and cathode will be present. So, see, this well, inside the well, the DNA isolated, we have isolated the DNA with restriction enzyme. That isolated DNA will be added into each well. 1, 2, 3, 4. You can find the well here. Okay, so what is the charge of DNA? What is the charge of DNA? Comment in the comment box. What is the charge of DNA? Okay, we have discussed this in the previous session. In the session, we have discussed with the structure of DNA, molecular basis of inheritance. Okay, what is the charge of DNA? DNA is always negatively charged. Okay, DNA is always negatively charged. So, it should be moved to anode or cathode. It should be moved to anode or cathode. It should be moved to anode, right? So, if this is negatively charged DNA, this is negatively charged DNA, and the charge here, the cathode will be connected here and anode will be connected here. Okay, so what happens? Sorry, the charge is in opposite side. Okay, so this is negatively charged side and this is positively charged side. Since the DNA is negatively charged, it will move towards the positive terminal. Okay, so how it is moving? See, you can find the thicker portion here and the smaller portion here. Okay, so we can say the DNA is moving according to its molecular weight, and also in further classes you have to study that it also depends upon the sieve size. Okay, so now we can consider the molecular weight. Okay, how the molecular weight? The highest molecular weight DNA will be retained there, and the smallest molecular weight DNA will be moving towards the positive terminal of the apparatus. Okay, so this is how we are separating the DNA. Okay, we are separating the DNA. After separation, it is being treated with ethidium bromide. Okay, why it is treated with ethidium bromide? To observe the DNA, it is being treated with ethidium bromide. After treating, we will move into the next process, that is elution. Elution means taking out the desired DNA. Okay, these are the different steps involved here. So let's say about isolation of derived DNA fragments. Okay, it is employed to check the progression of a restriction enzyme digestion. As DNA is negatively charged, it moves towards the anode. This is very important, dear students. It is very important. You have to study this. Okay, think about the structure of apparatus and the charges of DNA, then how it is connected, how well is prepared. Okay, after that you have to keep in remember that it moves towards the how DNA is negatively charged, and this DNA will be always moving to the which side? It moves to the positive terminal, that is anode. Okay, and this process will be repeated with the vector DNA also. See, now we are isolating the desired DNA. Vector DNA is also present, right? So this vector DNA also has to be specifically, it has to get cut and it has to get ligated. Okay, so after cutting the source DNA and the vector DNA, the cutout DNA DNA of interest from the source DNA and the cut vector are mixed and ligase is added. That means once everything is done after elution, here I didn't mention about elution, but you have to study about what is elution. That is also important. Elution. Okay. Elution. What is elution? It is the separation of DNA from the agarose gel. Okay. After that, you have to remember how the separated DNA. So if we have the apparatus as like this, see, if we have the apparatus, this is the first well, this is the second well, third well, fourth well. Okay. So this is negatively charged and this is positively charged. The DNA with different segments will be moving to different sides. Okay. Different sides according to its molecular weight. This you have to keep in remember. Okay, then what happens next? What happens next? My desired DNA is this. Okay, I have to get this as the desired DNA. After viewing it under the ethidium bromide, I have taken that, and then what happens? That separation of DNA, this particular DNA, is known as elution. Okay, that is known as elution. Clear? After elution, we got the DNA, desired DNA. Then what is next process? That DNA along with the vector DNA have to be mixed together. So I have a DNA here and also I have a vector DNA here. This have to get mixed. Okay, why it has to get mixed? It has to get mixed to form the recombinant DNA. Okay, it has to get mixed to get the recombinant DNA. So what actually has to be done here for the ligation purpose? Okay, it has the cut ends, right? If this is the DNA, if this is the vector DNA, the next target DNA or the desired DNA will be this. Okay, it has to get connected, right? How it can be connected? It can be connected with the help of enzyme called ligase. We have studied the ligase enzyme in molecular basis of inheritance, right? Ligase enzyme, Okazaki fragments. Have you remember about that? Okazaki fragments can be joined together with the help of ligase. Okay, so this is the condition for the next step, that is isolation of derived DNA fragments. So what was the first step? First step was to isolate the DNA. Okay. Second step was to cut the DNA. Cut the DNA with restriction enzyme. The third step is to isolate the derived DNA fragments. Okay. So next we have to move to the next step, that is amplification of gene of interest. Okay. Amplification of gene of interest. What is its sense? What does it means? Okay. Amplification of gene. So, as I have mentioned, first we can draw a cell. Inside the cell, what is there? Nucleus is present. Inside the nucleus, the DNA is present. After the process, we get our, see, this is the DNA. Okay, this is the DNA. Okay, so from this desired DNA, we get the desired DNA with the help of gel electrophoresis. Okay, we get the desired DNA. Then this desired DNA, it is then connected with the other DNA, that is the vector DNA. Okay, vector DNA. And now we get a recombinant DNA. Now what we get? We get a recombinant DNA. We will call this as a recombinant DNA. So this recombinant DNA, the only one DNA is not enough. Okay, only one DNA is not enough. For that purpose, what we are doing? We are doing PCR. Okay, polymerase chain reaction. Amplification have been done. That means multiple copies of DNA, multiple copies of recombinant DNA can be produced. Okay, multiple copies means not one or two or three, many a number of DNA can be accessed. Okay, can be produced by the process called PCR. What is PCR? It is polymerase. Okay, polymerase, it is the enzyme used. Okay, polymerase chain reaction. Polymerase chain reaction. important. It is also very important. See, I have already mentioned that gel electrophoresis is important. How the question can be asked after that, elution, ethidium bromide. Okay, that are the few things that you have to keep in remember for the exam point of view. Okay, next one, it is about PCR, polymerase chain reaction. You can expect either any of the processes from this chapter as a question. Okay, be thorough with gel electrophoresis and also be thorough with polymerase chain reaction. Okay, got it? So next we have to explain what is polymerase chain reaction and what are the steps involved in polymerase chain reaction. Okay, so we can say, see, you know what is the structure of DNA, right? What is the structure of DNA? It is double stranded in structure. Even it is recombinant DNA, it will be double stranded DNA. So for the process of replication, this have to be separated, right? That is the first step. Okay, separation of DNA. Okay, that is the first step. The DNA has to get separated. See, this is one DNA and this is next DNA. Okay, so that is the first step. Separation of DNA is the first step involved in PCR. Okay, then the second step. What is the second step? See, we have two different strands. Okay, so this what is happening in replication during after this separation of strands? Polymerase enzyme will be coming over there and the nucleotides will be added. Okay, right? That was the condition. Same condition has been applied here. Okay, here there is only one difference. The enzyme used for the replication process or the addition of nucleotides. Okay, it has a change and we will call it as tag polymerase. Okay, very important. Tag polymerase. Okay, what is tag polymerase? It is the enzyme used for the addition of nucleotides. My dear students, this tag polymerase is the enzyme which is being isolated from a bacteria called Thermus aquaticus. Okay, Thermus aquaticus. Okay, so the speciality of this bacteria is it can withstand above 190 degrees Celsius. Okay, it can withstand about 190 degrees Celsius. Okay, so from this bacteria, what is isolated? Thermus from this, what is the bacteria called? It is Thermus aquaticus. From Thermus aquaticus, what is isolated? Tag polymerase is the enzyme which is isolated. Why it is isolated? It can withstand 190 degrees Celsius. Okay. So, my dears, that is also one question. Why it is being isolated? Tag polymerase is being isolated from Thermus aquaticus. Okay. So, that is the second process. That means using tag polymerase, new nucleotides will be added. Okay. Since if new nucleotides will be added, what happens? The new DNA will be synthesized. Okay, that has been the second and third step. Okay, let's see what are the different steps involved in that. That is this one. Okay, first one, denaturation. It is a double-stranded DNA. This double-stranded DNA is separated. Okay, separated. That separation is denaturation. Okay, then what is the second step? The primers have to be added. What is primers? For a polymerase to be acted, first of all, there should be primers. Okay, with the primers only the replication can be started. This we have learned in the previous chapter, that is molecular basis of inheritance replication, right? So the action, after the action of primers, what happens? The polymerase can act. So second step is annealing with the help of primers. Okay, so first separated with the process is called denaturation. Second, what happens? Annealing takes place. Annealing with primers. Okay, annealing with primers. And then after annealing, DNA polymerase is added. What is the DNA polymerase used here? The DNA polymerase used here is tag polymerase. Okay, tag polymerase. Okay, tag polymerase is added. Then nucleotides are added. Why deoxynucleotides have been added into PCR? Does it has the ability to produce the nucleotides? Does it has the ability to produce the nucleotides? Yes or no? No, it doesn't have the ability to produce a nucleotide, right? But for the formation of nucleotide or formation of DNA, what is the must necessary condition or necessity? It is the deoxynucleotides. Okay, so we have studied that first it was a nucleoside. If we add nucleoside a phosphate group, then it becomes a nucleotide. The nucleotide then attaches together. Okay, nucleotides attaches together to form a polynucleotide, right? So first of all, the DNA has to be separated. Then the primer has to be added. Then the third step is polymerase is added. But along with the polymerase, what also should be added? The corresponding deoxynucleotides also be added. That is given here. Okay. Deoxynucleotides is given. Okay. After once deoxynucleotide and polymerase enzyme have been given, then the normal process of replication will be moving on. The addition of nucleotides will be taking place. A complete strand will be formed. Okay. And that process is known as extension. Okay, and the same three steps will be continued for how many cycles? 30 cycles. Okay, how many cycles it will be taken? 30 cycles. So around we'll get 1 billion times we'll get the DNA. Okay, one DNA we are providing one DNA, but after 30 cycles, around 1 billion amplified DNA will be obtained. So that much amount of DNA is now in our hand, not in our hand, in scientist's hand. Okay, fine. So see, that much amount of DNA is obtained. That DNA can be given to the vector organism. Okay, not vector, host organism. Okay, next let's see what happens after PCR. What is the step? Okay, so I told you there are DNA is isolated. Okay, then the DNA has been cut down with the restriction enzymes. After cutting the DNA, we have isolated it or separated it with the gel electrophoresis. The desired DNA, the exact particular DNA have been amplified using polymerase chain reaction. That are the different steps until now. Next, this amplified recombinant DNA. We have many recombinant DNA. We have many host cells. Okay, so we have to introduce this amplified DNA into the host cell. Okay, amplified DNA is there and this is a host cell. Then we have to insert this amplified DNA into the host cell. That is the next step. Okay, let's see what is involved here. We have already studied what is the importance of a host cell, right? We have also learned about the vector cell. What are vectors? Or usually vectors will be plasmids. Okay, then these vectors will be carrying the DNA to the host cell. Okay, why plasmids have been used in the previous class? We have discussed why plasmids. Plasmid, and what are plasmids, right? What are plasmids? Plasmids are the extra chromosomal DNA that is present inside a bacteria, right? So we can consider that bacteria can be given to any kind of host cells, or even in the bacterial cell we can take this vector DNA into the host cell DNA. Okay, or we can consider the bacteria as a host cell. Okay, fine. So, next process is the insertion of recombinant DNA into the host cell. Okay, so what happens here? There are several methods of introducing the ligated DNA into a recipient cell. There is a target cell. Okay, there is a host cell. There are different techniques or methods of introducing this DNA to this recipient cells. Okay, so, if we are considering the recipient cell takes the DNA into its body through its surroundings. Okay, we have also learned about the gene gun method. Okay, different method, biologistic methods we have studied. Next, if a recombinant DNA bearing ampicillin conditions, okay, these are the two different conditions applied. So let's see one by one. If a recombinant DNA bearing ampicillin resistant gene, okay, a selectable marker gene, it is transferred into E. coli cells. Okay, so we can consider a bacteria. Okay, we can consider a bacteria. This is a bacteria, and the bacteria has normal, not chromosome, it has a nucleoid. Okay, since it is not having the nuclear membrane, we can say that structure to be a nucleoid. Okay, so This DNA, it is, then there is a plasmid DNA also. We can say there is a plasmid DNA too. We have a recombinant DNA. Okay, and this recombinant DNA, we have a recombinant DNA. Okay, there is a recombinant DNA, and this recombinant DNA is being injected to the bacterial DNA. So the integration will be taking place. And what is the speciality here? The recombinant DNA bearing ampicillin resistant gene. What is the speciality of this DNA? It is ampicillin resistant gene. Okay, ampicillin resistant gene. Oh, if ampicillin resistant gene is transferred into E. coli cells, this host cell become ampicillin resistant cell. So this cell will be also ampicillin resistant cell. Okay, why? It has this ampicillin resistant genes. Since it has the ampicillin resistant genes, it can resist the ampicillin. Okay, the same condition is next given here. If the transformed cells are spread one on agar plates containing the ampicillin, only transformants will grow, non-transformants recipient will die. Okay, if it is the transformed cells are then spread to agar plate, again it is grown containing ampicillin. What happens? Those transformants will grow. Transformants means those which have been transferred after the insertion of the recombinant DNA will be survive. Okay, will survive. All the non-transformants, all the non-transformants means which doesn't take up the DNA. Ampicillin resistant DNA will not be survive. They will die. Dears, this also is an important part. They will give a twisted portion and they will ask the question for at least 1 mark question. Okay, that means sometimes they will ask in the case-based question, or else they will be asking this in a MCQ. Okay, this can be twisted in many ways. If ampicillin is given in the medium, how the bacterial cell, how the transformants will be surviving. That is one question. Okay, the same condition can be given with the tetracycline also. Okay, tetracycline. If the medium is having the tetracycline, will the transformant grow or will the non-transformant grow? Okay, so basically you have an idea about transformants and non-transformant. What are transformants? Those DNA which is having the recombinant DNA will get transformed. Okay, and that cell is known as transformant cells, and those cells which does not take up the recombinant cells in the medium is known as non-recombinant cells or non-transformant cells. That cells won't be surviving in the different conditions. Okay, fine. Okay. So, next one, obtaining the foreign gene product. Now, we have added the recombinant DNA into the host cell, right? So, we have to obtain the foreign gene product. How the foreign gene product have been obtained? The ultimate of aim of recombinant DNA technology is to produce a desirable protein. Okay, desirable protein have to be taken. Okay, so we know that DNA is present. Okay, DNA is present. If DNA is present after transcription, RNA will be formed. Okay, RNA will be formed after translation. After the process of translation, this is transcription. Okay, transcription. What is formed? RNA is formed. After RNA, the translation process will be taking place, and translation process will be helping for the formation of the protein. Okay, for the process of protein. Understood? Clear? So it is important. It is very important that you have to keep an idea on what is transcription, translation, and protein. So after giving the recombinant DNA, what happens? Transcription will be taking place, and that transcription will give the RNA, and that RNA will undergo the translation process to form the protein. Okay, so once we have to obtain the protein, and that is the next step, obtaining the foreign gene product. Once it is introduced into host cell, it is a foreign gene, right? Once is recombinant DNA is considered to be the foreign DNA. Okay, once it is added into the host cell, it will get integrated with the host cell nucleus or host cell DNA. Then from there we'll get the product. Okay, so that product, that is the next obtaining, okay, taking out the gene product as the next step. Okay, the foreign gene gets expressed under appropriate conditions. It will get expressed only with the appropriate conditions. That means the transcription translation have to be done with different conditions. Okay, if a protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. Okay, so what is a recombinant protein? If a protein encoding gene is expressed in a heterologous host. Okay, heterologous host means different host, not the same host. Okay, heterologous host, then it is called recombinant protein. Okay, heterologous host means we are isolating one thing. If you are isolating a DNA from one species of bacteria and we are giving it to another species of bacteria as host, considering another species of bacteria as host, if we are getting a particular DNA or not DNA, protein from the host, the another species of host, then we can call it as a recombinant protein. Okay, what is the condition here? It has to get expressed within the heterologous host. Okay, fine. The cells with foreign genes may be grown on a small scale in the laboratory. If it is a cells with foreign gene, it can be taken in the laboratory and also it can be taken from a wide space. Both the conditions are the possible. Okay, understood? Then the cultures may be used to extract the desired protein and purified using different separation techniques. Okay, so once the protein have been produced by the host organism, it can be isolated, separated, and it can be given marketed for the different processes. Okay, that is given. Let's move to the next one, that is downstream processing. That is another important step. Okay, downstream processing. What is downstream processing? Downstream processing means we get in the host organism, we get the foreign DNA product, that is the protein. Okay, this protein have to be isolated and it has to move for the next cycles. Okay, purification, isolation have to be done, purification have to be done. Then next, what is the next step? It has to get marketed. So all those processes have been involved in downstream processing. All the process to which, read the definition please, all the processes to which the product is subjected to before being marketed as final and finished products are called downstream processing. It is a question. Definition question. Okay, see what is a downstream processing? All the processes to which the product is subjected. What is the product? The product is the protein. What is the product? The product is protein. Okay, the protein. Protein. So this protein is subjected to before being marketed. Before marketing, what it has to, as I told, it has to get isolated. Then it has to get purified. Okay, that isolation purification steps involved is known as downstreaming process. It includes process such as separation, purification of products after the biosynthesis stage. So what are the two steps involved? This is the first step. Separation is the first step. Second step, it is the purification of products. Okay, then the products is formulated with preservatives. If the products is obtained, this products have to be maintained. This product have to be preserved, and it can be preserved with different suitable preservatives. Okay, clear? Such formulations undergoes through clinical trials as in the case of drugs. Okay, so once if it is a drug is isolated, drug is produced, what it has to be done? It has to get different clinical trials for an approval. Okay, so for that, for approval, it has to undergo the approvals too. That also comes under the downstream process. So strict quality control testing for each product is also required. How the quality is should be maintained, whether this quality is maintained or not have to be tested, and it is also done with the help of downstreaming process or under the downstreaming process. Okay, the downstreaming process and quality control testing vary from product to product. and all the process of separation, isolation, and the purification. And the next, what is the next step? Quality control. All this vary from different products to products. See, for example, we can consider the BT cotton gene. Okay, that the result is isolated and it is given in a particular way, but we are considering that with, if you are considering that with insulin, it is another case. Okay, insulin, what is the insulin? It is for all human beings, those who are suffering from diabetics, right? But BT cotton, that BT cotton can be taken and isolated, and it can be used for a further plants for prevent the pest organism. Okay, so the condition, according to the different condition, this downstream processing and the quality control methods will be different. It can vary from one particular product to the next particular product. Okay, fine, got it? Okay, so what is next? Bioreactors. Okay, this is an important part. This is the final part of our chapter about bioreactors. Okay, what are bioreactors? Let's see what are bioreactors. To produce large quantities of products, the bioreactors are used where a large volume of cultures can be processed. Okay, cultures can be processed. That means the if we have a target of obtaining 1 ton of product, okay, not just 1 kg or 100 mg. If you are planning to get a product of 1 ton, okay, then what have to be used? The bioreactors have been used. Okay, bioreactors. What are bioreactors? They are the vessels in which raw materials are biologically converted into specific products, enzymes, etc. using microbial, plant, animal, or human cell. Okay, what are bioreactors? Bioreactors, are the vessels. Okay, and what have been used in bioreactors? Raw materials are used, and what happens there? Biologically converted that products. Okay, the substrate, raw materials will be converted into specific products, enzymes, and it how it can be done with the help of microbial, plant, animal, human cell. Okay, a bioreactor provides the optimal conditions, temperature, pH, substrate, salt, vitamin, oxygen for achieving the desired product. We have to get a particular product, right? So, in order to get that particular product, all the optimal conditions have been given in these bioreactors. Okay. Then there are different types of bioreactors. Okay, so what are the two different major types of bioreactors? They are simple stirred tank bioreactor. Second one, it is sparked stirred tank bioreactors. Okay, the commonly used bioreactor is simple stirred bioreactor. So we have to study about stirred tank reactors. It is usually cylindrical or curved base to facilitate the proper mixing of the reacting contents. Okay, so just study that they are which is what is the shape? It is cylindrical in shape. Okay, stirred tanks are usually cylindrical in shape, and the curved base, here the curved base is present, right? So that helps to facilitate the mixing. Okay, mixing of the different molecules which is being given inside this bioreactors. Okay, and stirrer will be present, and the stirrer will be facilitating the mixing. Okay, mixing and oxygen availability throughout the bioreactor. What is the speciality of the stirrer present inside this bioreactor? It helps in the mixing. Okay, mixing and oxygen availability for the reactants to be converted into products. Okay, alternatively, air can be bubbled through the reactor. Okay, so simply we can say these are the important things that has to be present inside the bioreactor. Okay, if we consider a bioreactor, there should be an agitator. Agitator means which agitates. Okay, which agitates. You see the agitator in for the uh, purpose of taking out of the cheese, right? So same like there is an agitator system in the case of bioreactor. Then oxygen delivery, to supply proper oxygen, what is present? An oxygen delivery system will be present. Okay, then what is the third? A form control system is present. Form control, see, this is agitation. Okay, what is agitation process will be being taking place, right? So during this agitation, there is a chance of formation of bubbles. Okay, that means forms will be produced. So that form production have to be controlled, and for that there is a form control system. Okay, so next one, what is next one? A temperature control system will be there. Okay, for every process of agitation, what should be maintained? A proper temperature have to be maintained. See, my dear students, after working off an agitator for an even 1 hour or 2 hour, the temperature will be risen. Okay, it will arise. So, in order to maintain the temperature of this bioreactor, what should be present? A temperature control system should be present. Okay. So next one, pH control system. As you have studied in chemistry, pH plays an important role for the different chemical reactions, right? So a proper pH for the reactants have to be maintained for obtaining a proper result, right? So here also a pH control system is being practiced. Why it is being practiced? It is being practiced for the maintenance of proper chemical reactions to be taking place inside the bioreactor. Okay, so next, what is next process? Sampling force. Okay, for periodic withdrawal of the culture. In between what we have to take, we have to check whether it is converting or get converted or not. Okay, for that what we are using? We are using a sampling ports. Within the sampling ports, a small part of this sampling will be taken out. Okay, so what is next? The contents are mixed by stirrer. This makes the oxygen available throughout the bioreactor. Last simple step, single step. What is that? The contents which is being provided in the bioreactor, it is get mixed, it gets mixed well with the help of a stirrer. Okay, and also when the stirrer is working inside the bioreactor, you have to think that the proper oxygen availability will be also given. Okay, fine. So these are the different components, or these are different parts which is present inside a bioreactor. Okay, fine. Understood. Do you have any doubts? If there is any doubts, you can give in the comment box. Okay, about this chapter. So in this chapter, my dear students, once again, I am pointing out what are the things to be studied. See, what you have to study about the restriction enzyme. That is very important. Okay, restriction enzyme. So the images which is given in your textbook about restriction enzyme, also you have to think, remember with an example. Naming of restriction enzyme is important. Okay, what are the criteria used for naming a restriction enzyme? That is the first important part of this chapter. Okay, then within this restriction enzyme, you have to study about the palindrome sequence. Palindrome sequence. Define how to define this. Okay, you have to study this palindrome sequence. Okay, next you have to study about vector with example as PBR 322. Okay, these what is a vector? PBR 322, the image, the diagram. You have to study the diagram. Okay, then next you have to study about this different, these are the tools. Okay, next the different steps involved. This you have to study for 5 marks. Okay, steps involved in DNA recombinant technology. For 5 marks you study this. Okay, then for 3 marks, my dears, you have to study about, this is for 5 marks. Okay, 3 marks you have to study about gel electrophoresis. Okay, gel electrophoresis, and also you have to study about the second one. It is about what? Polymerase chain reaction. Polymerase chain reaction, and also you have to study the diagrams. You have to study the diagrams. Okay, these are the most important possible questions from this chapter. Along with all others are important, but for sure you have to study this and you have to go for your examination. Okay, don't leave anything, but give priority for this questions. Okay, and also the last step, I think that is bioreactors, right? That is also downstream processing. How to define downstream processing and what are bioreactors? You have to remember that too. Okay, so we are going to wind up the session, and next session we will be continuing in the coming classes. Okay, so next class or next section we will be continuing with question and answers or like a marathon. It will be easy for you to understand how to write the examination. Okay, so all the very best for your upcoming exams. Write it well. All the very best from GoScore family. Thank you.